So first of all, Sadik, thank you for being here today. It's my pleasure to have you as our subject matter expert for our first Cell and Gene Live event. So I appreciate you and your time.
Yeah, thanks for having me, Erin. It's a pleasure to be with you today.
Absolutely. Okay. So let's get rolling. First of all, the introduction of CRISPR technology and the ability to grow stem cells in the lab has been among probably one of the most important breakthroughs in science, right? So, but before we kind of get to the advancements, set the stage for our listeners for say 3 to 5 years ago regarding CRISPR and stem cells. What did CRISPR engineered stem cells? What did the therapy look like back then?
Yeah, absolutely. And maybe is it okay if I go even beyond 3 years, maybe even earlier? Um,
Absolutely, absolutely.
you, so I think it's important to start off by saying that CRISPR Cas9 is a gene editing tool. So it enables us to copy and paste parts of the human genome with a therapeutic purpose in the end. But if we look at gene editing tools, they've been around for quite a bit. There's at least three classes of gene editing tools. These include things like zinc fingers, homing endonucleases, and CRISPR is the most recent iteration of gene editing tools. And really, the excitement around CRISPR really evolves around the fact that it's democratized the concept of gene editing. It's enabled in a very flexible and cost-efficient manner folks to edit parts of the genome, and it's been applied in a variety of contexts, including agriculture, diagnostics, and therapeutics. But if we look at CRISPR, although this year the Nobel Prizes were awarded to Emmanuel Charpentier and Jennifer Doudna for their seminal discoveries back in 2012 about how CRISPR can be used to modify single cell organisms in terms of their genes. The actual discovery of CRISPR as a concept dates all the way back to 1987 when it was initially discovered in bacteria as an immune mechanism for them to eliminate viral infections. So, really the point that I'm trying to make here is that the concept of gene editing has been around for at least 3 to 4 decades. Within that, CRISPR has been around since at least 1987, but its first potential application or demonstration of a use case happened in 2012 with the work of Fang Zhang, George Church, Emmanuel Charpentier, and Jennifer Doudna. So since 2012, about 8 years ago, there's been a tremendous amount of progress, which is almost unprecedented in clinical development. How quickly we've gone from the proof of concept in a petri dish all the way to therapeutic applications. And I would say, to answer your question, what was the CRISPR therapeutic landscape 3 years ago? The answer is that there was really no trials to speak of. It was still the beginning of a lot of work enabling potential clinical application. But 3 years ago, there still had not been a single patient treated. Now, since that time, if you go onto clinicaltrials.gov, I did so this morning, there's at least 42 different trials or potential trials that are open that are evaluating the application of CRISPR gene editing within the context of therapeutic and clinical applications. And some of the data is really emerging. It's looking really exciting, really promising that in certain applications, CRISPR gene editing could lead to potential curative options for diseases where there currently are no curative options. And we can get into some of those studies. So to summarize, the concept of gene editing has been around for at least 3 decades. Therapeutic applications of CRISPR gene editing are more recent and have only begun to emerge in the past 3 years. And this year, is really an important year because we've gotten the initial readouts of some of those initial clinical trials for CRISPR gene edited therapies.